two, one.
Continuing on with translation. So what we see is we have these codons, these triplets of nitrogenous bases within the mRNA. So we've done our transcription. We've gone from our DNA template. We've made our RNA using those pairing rules where T will pair with A with U, C and G back and forth to make our RNA. And now these codons, these sets of 3 nucleotides, are going to code for a very specific amino acid. And so these tables are how we figure this out. And on your quiz, you will have this table available when you have to do this process of translation. So we've got our AUG here within our mRNA. So we find AUG on this table, and that gives us methionine. Okay, and you can just write met. Then you look at the next codon, AAG. Okay, and so we find that on this table, AAG for lysine. So you write LYS. Then we look at our next one, UUU. So we find our UUU here, and it's phenylalanine. And so you write the PHE. And then you continue on depending on how long that mRNA strand is. And this is how we get that primary structure of proteins. Okay? It's just copying these mRNA, translating it into amino acids. Okay? There are 64 different codons, 61 are for amino acids. The last 3 are for stop codons. When we get to these stop codons, that says we're done, the protein is finished. Okay, we see there's a lot of repeating here because there aren't 61 different amino acids, there's 20. And so this repetition can actually be good for the cells, for the DNA, because it helps to make sure that if there's a mutation, it's possible that it won't do anything, because if we get a mutation that changes this codon from ACU to ACC, it's still the same amino acid, so it doesn't change that protein. Okay, so these are the steps of translation. Okay, so we get mRNA. It's going to leave the nucleus, enter the cytoplasm. Specifically here, we are at the rough endoplasmic reticulum. This protein synthesis is going to happen at the ribosomes. So this mRNA is going to come over, and it's going to pair itself up with a ribosome. So that ribosome, or that mRNA, is going to make its way through that ribosome until it gets to a start codon. At that start codon, it's going to bring this tRNA molecule. This is transfer RNA, and this carries the amino acids to the ribosome. What we see with these tRNA molecules is they have an anticodon that's going to match up with the codon. So our start codon is AUG. The anticodon on the tRNA is going to be UAC. And so this is how the ribosome knows which amino acids to use, because the tRNA is going to have a code that's going to match up with the codon on the mRNA. And it has a specific amino acid attached to this tRNA. So we find that start codon, it brings in methionine. Then we move on to the next codon on the mRNA. Another tRNA comes in holding a different amino acid. And so what we see is, again, we get this methionine to come in. We've got the next code here. Leucine's going to come in. These are going to bond together, and then we are going to shift over where these tRNAs are. So the one from the A site is going to move to the P site, and we're going to see this here. Okay, we get a bond between these amino acids, and then we're going to get some shifting. Okay, that tRNA that dropped off that methionine is going to leave, and now we're going to move from the A site to the P site with this tRNA. Okay, and then we're going to move to the next codon, and the tRNA with the anticodon matching it is going to come in with a different amino acid. We're going to get a peptide bond, and then we're going to shift again. Okay, so we're going to shift over, move to the next codon. tRNA matching anticodon to codon is going to come in. In this case, it's alanine. We're going to get another peptide bond. And this process is going to continue, bringing in new tRNAs, getting these peptide bonds, shifting over, new tRNA, and so on, until we hit a stop codon. And then we have the primary structure of this protein that we've translated from the messenger RNA. Okay. And then it goes through this process of folding, right? We have to, different parts will form the beta sheets or the alpha helices, and then we'll get different polypeptides together after they fold three-dimensionally so that we can get that tertiary and quaternary structure of that protein. Okay. So looking over this central dogma, again, the central dogma was the process of going from DNA to RNA to protein. Looking at replication, where does it happen? In the nucleus. The template for replication, so what are we copying from? DNA. What are we making? More DNA. What enzymes do we have? Polymerase, specifically DNA polymerase, our DNA helicase, and then also ligase. Okay. Transcription, where does it happen? In the nucleus, right? Because we're copying from DNA, and DNA always stays in the nucleus. What are we producing? Messenger RNA. What enzymes are involved? We've got RNA polymerase. Translation happens in the cytoplasm. The template is our messenger RNA. We are building a protein, and we're using the ribosome. Okay? And so that's how we're getting this process done here. Okay? Now, it is important to note that not all of our genes are always functioning. We've got some that get turned off and turned on, and it's all based on these promoters, operons, these regulatory genes that can turn these genes on, turn them off. And this happens throughout our lives, where various genes are turned on, turned off, depending on a lot of different factors. Okay. Gene regulations, we can repress genes so we can turn them off, we can turn them on, express them, or we can ramp them up so that they're really producing a lot of those proteins or whatever they code for. Mutations. Here we've got a normal fruit fly with its big old eyes. Here we've got a mutant fruit fly where those eyes formed very differently. Okay, and this is because of some mutation, some error in the DNA. Okay, so mutations can be harmful or fatal. Okay, they could kill an organism, cause some disease. They can also have no effect, as we looked at with codons. A lot of the times, if you change a single base, it's not going to affect the protein that's made. It's going to give us the same exact amino acid. They can be beneficial. This is the whole idea behind natural selection. Sometimes these mutations give us some sort of advantage. Okay. Now, I will say that it is rather rare, okay, but it does happen because there are millions of organisms and millions of mutations that occur. It's bound to have one be beneficial at some point. We've got various types of mutations. We have a base substitution. This is where you just change a single base. Okay, so here in our example down here, we change one base within this DNA, which changes that amino acid. Okay, can be bad. Sometimes it might not make a huge difference. Okay, but that's a base substitution. You're changing one base. Now we can get a missense mutation, which is a type of base substitution, and this is true for the disease sickle cell anemia. What we see is it changes a single base, and this causes this disease because hemoglobin isn't able to form correctly. Okay, so we'll stop here at base substitution, and we'll finish up with mutations in the next video.